All right, everybody, let's start with we are going to be learning about viruses. Here we are. So, viruses is what we're going to be going into. This might take more than one video. Let's see what happens. And if you have any questions at all, please contact me. All right, so our learning objectives. Let's talk about what are the things that we want to make sure we learn and understand. So, first thing, explain how viruses reproduce. How do we get more viruses? How do we replicate what we have? Describe what happens after a virus infects a cell. So, what happens after that virus gets into the cell? 3. Explain how viruses cause disease. 4. Clarify whether a virus is a living thing. So, have you ever missed out on something important or even just school because you had a cold? Well, a cold is an example of an illness that is caused by a virus, and it's not always the same virus when you say you have a cold. So, this lesson is going to describe the structure of viruses and explain what happens when a virus infects your cell. So, we have a little bit of vocabulary. If I were you, I would pause. I definitely want you to write this down. You know how I am about vocabulary, so go ahead and take out your notebook and write these down. The first four are virus. Next is capsid. Next is a lytic infection or lytic infection. And fourth is bacteriophage. Now, let's start off with virus. This is listed as a particle made of proteins, nucleic acids, and sometimes lipids that can replicate only by infecting living cells. There are a few words that you should really know, and they are called your biomolecules. The only biomolecule missing from this list is carbohydrates. So remember last semester we talked about carbohydrates, lipids, proteins, nucleic acids. Three of those are literally in the definition of virus. If we go to the next word, capsid, this is a protein coat surrounding a virus, meaning it's going to be a surrounding coat surrounding a virus. Like it surrounds it, it encapsulates it. Next, we have lytic infection. This is a type of infection in which a virus enters a cell, makes copies of itself. Ha, it says comma. Let me fix that. It makes copies of itself and causes the cell to burst. So when you hear lytic, you should think burst. Bacteriophage. Well, the beginning of the word sounds like bacteria, so we know that it has to have something to do with a bacteria. It's also been called a bacterial virus, and the actual definition of it is kind of infection that infects bacteria. All right, let's move on to the next slide. You can always pause the video so that you can write down whatever you need. Your next set of vocabulary, you have lysogenic infection, prophage, and retrovirus. With a lysogenic infection, when you see something that says lice in front of it, that means that something is there's going to be destruction of something, right? So this is going to be the type of infection in which a virus embeds itself, meaning like it inserts itself. It becomes a part of its DNA. So the virus embeds its own DNA into the DNA of the host cell and is replicated along with the host cell's DNA. So As you have cells that are having their DNA replicated every single time there's a new cell, the virus has embedded itself into that DNA. And every single time you have replication of a cell and replication of your DNA, you are going to replicate this virus. Crazy, right? Now, the next word is prophage. This is going to be a bacteriophage. And if we look back, we just learned that word, bacteriophage, a bacterial virus. So a prophage is a bacteriophage DNA that is embedded in the bacterial host DNA. So again, we have something that's being put into the host DNA. The last word we have is retrovirus, RNA virus that contains RNA as its genetic information. So we have DNA as our genetic information. A retrovirus has RNA as its genetic information. All right, so let's talk about the discovery of viruses. In this picture, you have the tobacco mosaic virus, and it's showing you that there's one single strand in there, so you know that that's RNA, and then it's also labeling the capsid proteins. But right now, we're kind of focusing on the discovery of viruses. So, first one, in 1892, Dmitry Ivanovsky, he demonstrated that the cause of tobacco mosaic disease was found in the liquid extracted from infected plants. So they actually took out the liquid from an infected plant, and that's how they found this. The second one, all right, I'm trying not to butcher it, Martinez Bierjanic or Bierjurinic. Hopefully I got it. If I didn't, at least I tried and I attempted it. In 1897, he suggested that tiny particles in the juice cause the disease. He named these particles viruses. Here's an interesting thing. Viruses is termed after the Latin word for poison. So the word viruses or the particle viruses that he was using is for the Latin word poison. In 1935, we have Wendell Stanley. He isolated crystals of the tobacco mosaic virus. Now, just as a quick recap, in a little bit, I'll show you the picture of what you guys should have seen on your homework from last week. All right, here we have, it says virus structure and composition. That means what kind of shape does it have? What is it made of? That's what this means. So again, capsid is going to be a protein coat surrounding a virus. Here in your T4 bacteriophage, in the T4 bacteriophage, you have a head, which houses the DNA on the inside. You have the tail sheath, and then you have the tail fibers. If we look at the next one, and actually right here, we also have this microscope, and it's showing that it's at 60,000 times the view. That means it needs to be very, very small. This has to be extremely small. All right, now the next one is the tobacco mosaic virus. Same as the picture before, you'll notice that it has the encapsulated proteins and the RNA coil on the inside. If we're looking in a microscope, this is what it is looking like in the microscope. No, it's not going to show in color, but you know, when people actually put these together, they add color to it so you can decipher between what's what. If you look at the microscope, this is actually zoomed in for 40,000 times. So this one is a little bit bigger than this one. And then if you also notice that the virus looks similar to the tail sheath of the T4 bacteriophage. So this right here looks very similar to this. but it does not have the head and the tail fiber of the bacteriophage. Also, the size is way bigger because if you were to zoom in on something at 40,000 times zoomed in, and then you zoom in on something that needs to be seen at 60,000 times, this one is clearly smaller. All right, and then this last one is going to be the influenza virus. This is what you guys know as the flu. This virus looks nothing like the other two, but it should look familiar from the website that we looked at with the virus exploration. This T7 virus is very similar to what you just saw here at the T4. It has the head, the tail sheath, and the tail fibers. So this would be your head. You can't really see the tail sheath, but if I would have turned it around, you would. If you look right here, you can kind of see what it looks like. And then it has these little legs going on right there. All right, moving on. This tobacco mosaic virus, we just talked about this. This one comes from the Virgaviridae family. Ooh, hard, right? We also know as linear, uh, single-stranded, positive, and it's an RNA virus. We know all of that just from this part. You should have seen this on your homework. And the last one that you should have seen on the homework was the influenza A virus. And it's just kind of giving you this information. If you didn't get to this part yet, I suggest you definitely go through it. It has an amazing amount of information on there. You can look at the 3D model. If you're looking down here, you can turn it this way, you can turn it sideways, you can flip it around, you can click this button where it says cross section, you can cut it in half, and you can see what's on the inside. So definitely take the time to look at some of those things. If we're looking again, what type of nucleic acid does each virus type have? So what type of, that means, does it have DNA or does it have RNA? We know that there are two types of nucleic acids from what we've gone through already. And again, DNA versus RNA. This T4 bacteriophage has DNA, so we know that this is double-stranded. This one has RNA for the TMV, and influenza or flu has RNA as well. So that is how we know what type of nucleic acid or one of the biomolecules that each virus has. We're going to skip ahead to where we just came from, and boom. All right, so a virus is a non-living thing. A virus is a non-living thing. So American biochemist Wendell Stanley is the scientist who isolated or separated crystals of the tobacco mosaic virus, or TMV. He knew that living organisms, they don't crystallize or turn into like crystal shapes, right? So he inferred that the viruses were not truly alive, because if you have properties that living things can't have, it's not alive. So a virus is a non-living particle made of proteins, nucleic acid, and sometimes lipids. How do viruses reproduce? Viruses can only reproduce by infecting living cells. This is the first part of your notes for this slide set for chapter 21.1 of viruses.